So today I'll be talking about some spatial statistics we're using to try to understand local adaptation and spatial patterns in ancestry, and then also how we can maybe try to apply some of our knowledge to crops. So for a long time, about as long as I can remember, I've been interested in spatial pattern. I think a lot of people are interested in spatial pattern, and it's partly because of how our sensory systems are geared. We're very good at recognizing spatial patterns. And, for long periods of time now, scientists have tried to understand the processes that underlie different spatial patterns. And it's often one of our most important tools for trying to understand what different processes might be acting in a given system, that is inference based on spatial pattern. We're also interested in spatial processes that change through space, that are conditional on spatial location. And in evolutionary biology, the two areas where I'm interested in thinking about these kind of big issues is in trying to understand patterns of gene flow across landscapes and continents, and how demography and changes in populations through time and space can influence patterns of relatedness and diversity. A lot of our work is with sorghum, the global crop that has spread from sub-Saharan Africa, where it was domesticated, to a much wider range. We're also doing some work on some invasive species and trying to understand patterns of population genetic diversity over time. And then more specifically, with regards to adaptation, I'm interested in understanding local adaptation. It's often the case that researchers will study common garden experiments and look at traits that they think might be important for local adaptation. And if we see genetic variation in these traits that corresponds to environments of origin, then we may hypothesize that this variation is under spatially varying selection involved in local adaptation. And so one of the things I'm interested in doing is trying to understand the genetic and physiological bases of these locally adapted traits and how they contribute to adaptation. And then what I'm interested in doing is taking some of what we think we're learning from population genomic scans for local adaptation all across the genome, trying to find loci that are locally adapted, and trying to apply that knowledge in some way. Whether it can be useful or not, I think is still an open question, at least in terms of practice.
Jesse,
yes.
Sorry for interrupting. We are still on the first slide. I'm not sure.
Oh, no. Oh, no. Specific questions that I'm going to address today are first trying to use a new tool. It's not exactly new, but we're implementing it in a new way, that is applying wavelet transforms to spatial pattern and allele frequency to try to understand first patterns of relatedness across the genome in space, and then also to identify specific loci that might be locally adapted. And then secondly, I'm going to think about maybe some applications of learning about the genetic basis of local adaptation in cropland races, that's local varieties. We're going to do that in sorghum. Okay, to start out, I'm going to talk to you about this new tool we're trying to develop. And we're going to apply it both for trying to understand spatial patterns at a single locus and also across the genome. And we're going to do this to both try to get at the basis of local adaptation, but also to understand something about patterns of gene flow across landscapes. And one of the reasons that I've gone down this path and something's motivating me is that oftentimes when we're trying to build statistical models for change in allele frequency across space, it's not exactly clear what kind of functional form and at what spatial scale that variation might be occurring. If we're thinking about declines in allele frequency, either in space or on environmental gradients, they could be very sharp and nonlinear, or they may be more gradual and closer to linear. And we often find too that genome-wide, the ancestry turnover is not smooth in space, but it can be sharp in some regions, perhaps smoother in others. And so what we want is something, some analysis tool that is flexible to accommodate this variation. In the case of local adaptation, we often don't know specific environmental gradients that might be driving local adaptation. Depending on your system, there's many potential options. And one of the tools that people often use that can overcome this issue is looking for allele frequency differentiation among populations, where you don't necessarily have to know what it is about the environment that differs between places. But there's a challenge associated with this that I'll come back to in a second. And it's a challenge shared by another nice approach, though. So this is the estimated effective migration services approach, which takes samples from across a landscape and puts them into a gridded set of subpopulation bins, and then infers local migration rates between cells on the landscape, essentially. The challenge, though, that motivated me thinking about both of these types of approaches is that we don't always know where we should be discretizing populations in space. What is the subpopulation that will allow us to capture the process we're interested in is not clear. And what happens if local adaptation or changes in population structure are also changing across spatial scales, so that it's not always something that's happening at a consistent spatial scale. So let me sort of lay out how this might play out in sort of calculating FST among populations. In the ideal world, if we knew what the selective differences were between locations, what about it was the environment that was driving that, we could potentially delineate our subpopulations accordingly. But if we don't, we might put a grid down on the landscape, and that grid may or may not match up with the actual environmental differences that drive local adaptation. But maybe we could slide that grid across the landscape to avoid any problems associated with where we've anchored the grid, or problems associated with aliasing, as it's known. So you could slide that, and maybe sometimes the grid matches better than other anchorings, the actual selective differences among populations. But if the spatial scales at which the variation is occurring is also changing, perhaps this should be implemented in a multi-scale fashion. So you have different sliding windows of different sizes across the landscape. And this is sort of a crude analogy to what we're doing with wavelet transforms. And I'll describe those in more specifics in a moment. So my other co-authors on this study are Tim Kitt at UT Austin and Diana Gamba, who's a postdoc in my group at Penn State. And the work that we're doing here partly builds off an implementation that Tim developed earlier for characterizing spatial turnover in ecological communities, where the analogy here is allele frequencies across the genome. So this is just sort of a little cartoon example of how the wavelet filters that we're using work on spatial allele frequency data. So this is a linear, one-dimensional landscape on the x-axis there. You'll see at the top that there's this fluctuating allele frequency where you sort of got 3 patches with high alternate allele frequency towards the center of the landscape, and the other allele or the reference allele on the rest of the landscape. And what we can do is take a wavelet filter, which is a function that has this localized character to it, and we can apply that filter to the allele frequency data. That is, take the product of the two, and then we can ask how much variation do we see in the wavelet transformed allele frequency at a given location and at a given scale. And the wavelet that we're using here is a difference of Gaussians. This is subtracting 2 different Gaussian functions. And S gives the dominant scale of variation in that filter, and A is the location where it's centered, and A is 0 here. So when you take this approach to these allele frequency data on the top there, then you look at how much variation is there in the wavelet transformed allele frequency, you end up with 2 peaks across scales. So this is at far right here. We've got a peak around the scale about 0.18. And so that means there's a lot of variation in the wavelet transformed allele frequency. And if you plot that out, plot out the filter, which is this black function here, and the allele frequency data, you see that it matches very nicely the scale of oscillation between these small patches in the middle of the landscape. And then when we go out to larger scales, we get another peak that's around the scale of about 1.64. These are just in units of this arbitrary landscape. And at this scale, what we see is a very wide dilation in the wavelet that is characterizing the fact that this alternate allele is more common in the middle of the landscape than it is at the edges. It disappears at the edges. And so this, applying these filters at different scales allows us to pull out signals that are occurring at different scales in the allele frequency data. So to give you something a little bit more concrete about how this works, we have the difference of Gaussian's wavelet filter. I'm not going to dissect everything that's in it, but I showed you the forms, what those look like visually in the previous plot. And those are multiplied by the allele frequencies at a given location. And then that's integrated across space, that allele frequency. And then that gets normalized depending on the variation in the wavelet filter at that scale. So what we then get is, for a given location, a wavelet transformed allele frequency in a given scale. It essentially says how, what is the frequency of either allele at a given scale of analysis, sort of smoothed across space essentially, and centered at an individual location. Now, to get at genome-wide patterns of genetic similarity, or in this case, dissimilarity, what we can do is take the combined distances, turn those into distances essentially, and get a what is essentially a Euclidean distance in the wavelet transform space. So this is aggregating across loci, which are indexed with the I here. So what we then do, I'm going to start out showing you here are some individual-based simulations using the SLIM software. So these are using our life history that is approximately something that is outcrossing and short-lived perennial, when I'm thinking about plants. And some of the simulations below will have some spatially varying viability selection. But start out, they're going to be neutral where there's no selection. Okay, so let's look at some geographic patterns and relatedness. Now, the sort of simplest example is this 2D continuous landscape. The whole landscape can be populated. After a long period of time, you get fairly smooth patterns of population structure across the landscape. I'm just plotting colors here to indicate the first two principal components of the SNPs. And you can see that it's pretty smooth, and the two PCs essentially tell you where you are in the landscape. And then one of the things we can do to sort of get a sense of what is going on is we can take across the landscape the mean wavelet genetic dissimilarity at a given scale and compare it to a null that is based on permuting the locations of the individuals, which is essentially what you would have with panmixia. And so you see at these smaller scales, there's less genetic distance than expected because things that are nearby each other are more closely related than expected under panmixia. And then at larger scales, they're more different than expected. So that's just sort of trying to give you a little bit of intuition for how this works. Okay, so let's think about a heterogeneous landscape where we have lots of patches that are suitable and some that are unsuitable, maybe like islands, let's say. So we, again, simulated some neutral evolution across this heterogeneous landscape. In this case, the little gray squares on the figure in the middle there are patches of suitable habitat, and the white are places where individuals will perish if they land there. You can think of that as an ocean maybe, and this is a terrestrial organism. So this, what I'm showing you here, each circle is an individual sampled from across the landscape. And we calculate the wavelet dissimilarity centered at that individual's location at a given scale. And the colors are indicating the relative variation in that wavelet dissimilarity. And because they're blue, that means they're all significantly less dissimilarity than expected. That is, nearby genotypes are more similar than you expect. But one thing I want to point out is that we can already see some strong spatial variation between parts of the landscape in terms of the level of low wavelet dissimilarity, or wavelet similarity, if you want to think of it like that. And what you see in the upper left here in this sort of isolated island in the upper left, and this also happens for this island here as well. These things are small, isolated. They have much greater or much lower dissimilarity at small spatial scales. This is the scale of analysis here, about 2 distance units. They have much lower dissimilarity than expected at that scale. So we can look at just a plot of geographic distance versus genetic distance pairwise in this region. And you can see it only increases a very little bit as you get out to this kind of genetic distance. Things are still very similar to each other. Whereas when we look in the middle of the landscape, you're getting a much greater increase in the genetic distance with space. So there's higher dissimilarity in that region, although in many cases still significantly low. Now we can look at some larger spatial scales for the wavelet dissimilarity. And we get a flip in the pattern here where these island populations that are isolated in the upper left and lower right, these have significantly high wavelet dissimilarity and the greatest across the landscape. And to sort of give you some intuition for what this is picking up, this is identifying the fact that these populations in the upper left and lower right are very distinct from populations that are separated from them at this scale. So this is at this larger scale of analysis. And you see pretty much a similar pattern as we go to larger scales. And just to show you, if you do a PCA on the SNPs from this simulation, the individuals from this sort of mainland population, if you want to think of it like that, all cluster in one part of the PC space, and then each of these island populations cluster in a different part of the PCA space. So there's not a lot of migration across this open water here. It's pretty rare. And so that gives you this greater wavelet dissimilarity at this larger scale. It's still significant in the middle part of the landscape, but it's not as great because there's presumably more gene flow across this continuous continent, let's call it. So let's look at this in some empirical systems. Two things that I work on a lot, Arabidopsis thaliana. So we're working with 1001 genomes resequencing data here and populations from across Eurasia and a little bit of North Africa. And then sorghum landraces. Landraces are traditional varieties of crops that are local. So they're thought to be adapted to local conditions, and experiments often show that. But this in sorghum here is very broadly distributed. It's domesticated sub-Saharan Africa, but it's spread as far as northeastern China. And when we apply this wavelet dissimilarity and then compare it to that null expectation from panmixia, one of the things that pops out to me that is interesting between the comparison of these two is that sorghum shows significantly low dissimilarity up to much larger spatial scales than does Arabidopsis. So up to about 50 kilometers, sorghum are still less dissimilar than expected compared to the panmixia null, whereas for Arabidopsis, it's only at a few kilometers. And once you get up to 50 kilometers, they're well more differentiated than expected. So Arabidopsis is largely selfing like sorghum, but there's seed dispersal is very little sort of seed dispersal mechanism in terms of any specific adaptation for seed dispersal. Seeds are sort of just very small and fall to the ground. Whereas sorghum, we think farmers have been sharing sorghum with neighboring farmers for long periods of time. So this would seem to indicate a larger scale of gene flow across the landscape. And then we can also look at the individual locations that are sampled across the whole study globally and look for regions where we see distinct patterns. And one of the things that we pick up for a still fairly small scale, where sorghum actually were more significant or less significant, less dissimilar than expected. The Arabidopsis, we see a few regions that stand out. Northeastern Iberia is significantly more dissimilar than expected at that scale, also northern Sweden. And one of the things that's notable about this region for Arabidopsis in Iberia is that this corresponds to the region of greater topographic heterogeneity. So if you look, all of these locations are where the Arabidopsis at this scale already starts to show significantly high dissimilarity at these individual locations. And it's all pretty much associated with these major mountain ranges of Iberia, which we take as evidence that there's limited gene flow because of either selection against immigrants if they're locally adapted, or simply less movement across these steep landscapes. In sorghum, looking at a bit of larger scale, about 260 kilometer scale, you see a number of parts, mostly of the sub-Saharan part of the range in South Asia, where you have significantly greater dissimilarity than expected. One thing that's interesting to me, though, is here in China, we have, even at this fairly large scale, significantly low dissimilarity. And this is the region of sorghum cultivation that's colonized most recently. And so there's sorghum cultivation in this region has only been there for 1,000, 2,000 years maybe, so much more recent than the rest of the range. So what that's telling us that these are much more similar across large distances than expected. Okay, so let's transition a bit now into trying to find locally adapted loci in a genome scan. We're going to build here on some work that was done by Lauterhaus and Whitlock, or Whitlock and Lauterhaus, and also Lewontin and Krakauer, going back further, where the idea is to fit the statistic to a distribution of statistics and to use that as a parametric test of whether there's a particularly high test statistic value. in the case of FST, what Whitlock and Lauterhaus did was show that you could use the observed distribution of FST values with some trimming at the tails to actually fit the degrees of freedom to the chi-square distribution, because in the case of a heterogeneous metapopulation network, the number of degrees of freedom of that distribution is not clear. It's not immediately obvious. And so what we're going to do is essentially create an analog for wavelet variance, where we expect it to still follow chi-squared because it's a sum of squares distributions, and standardizing it by, so we're taking the wavelet transform at a given locus and standardizing it by the allele frequency variation, or the standard deviation of allele frequency at that locus, and then taking the variance of those standardized allele frequency transforms. And we're going to apply that to, in this case, I'm showing a simulation of local adaptation to a distinct environmental patch, this square in the middle of the landscape. It's different from the rest of the landscape. And we can see in this case from our simulated data that the scaled wavelet variances follow the chi-square distribution pretty nicely, and we're using the maximum likelihood approach of Lauterhaus or of Whitlock and Lauterhaus to actually fit this chi-square degrees of freedom. And then we use that to calculate p-values from the upper tail of individual SNPs. Now, this is probably an obvious point to people, but I just want to make the maybe consider a straw man comparison here, but be it as it may, if you don't define the borders of the population as well, FST will probably not do a good job of finding the locally adapted locus. So in this case, where it's really defined about as poorly as you could do, this single causal SNP that is underlying local adaptation is not identified as an outlier. It is to a greater degree in this case, we've done a little bit better of a job, but that's just to sort of make the point that it can be challenging if you don't delineate your subpopulations right. So in the case of applying this scaled wavelet variance test, on the top I'm showing you the actual scaled wavelet variance values. And one of the things that I think is useful from this is that the actual wavelet variance, that is the variance in the wavelet transforms, can be pretty high at larger scales. So the red circles here are larger spatial scales. The blue circles are smaller spatial scales. That wavelet variance can be pretty high at some of the larger spatial scales, but those are neutral peaks. Those are loci that are not causal for the trait under selection. And so those appear to be just loci that show strong differentiation because of limited gene flow across the landscape. But when we calculate the p-value using the chi-squared distribution approach, we see pretty good resolution here of that locally adapted locus. And just zooming in here, I'm just showing you the colors of this, that is indicating the scales at which the p-values were lowest for this test. Those are sort of intermediate scales. So what that is indicating to us is the power for this analysis is greater in intermediate scales, where maybe we have less confounding due to limited gene flow. That is, fewer cases where you have very high wavelet variance because of limited gene flow at those intermediate scales, but where you still have difference in environments selecting for different alleles. Okay, so we can also apply this to the Arabidopsis 1001 genomes data. And the top QTL, a couple SNPs down here on the list, comes out at DOG1, which is a locus that controls phenology in Arabidopsis, where there's been several papers demonstrating some of the details of how this works on a molecular level. One of the things that's kind of interesting about DOG1 to me in this analysis is that we in my group and some others have tried to understand the environmental pattern and the variation at DOG1. And one of the sort of confounders is that the environmental clines in phenology in Arabidopsis change from region to region. And so, for example, the correlation that DOG1 has with elevation and temperature in the Western Mediterranean is different from what it is in other locations. But what the wavelet test is telling us is that this spatial pattern of allele frequency variation is very non-random, and so much so that it stands out across the genome. Here's a larger scale SNP that came out of the locus of interest, just to sort of give you an idea about the kind of patterns that we're able to pull out. Here's applying those data to sorghum. At the largest scales here, we see at this glutathione S transferase, we pick out the SNP across the genome that is nearly fixed, comparing Eastern and Southern Africa versus Western and sort of Sudan and Southern Sudan. So that's sort of trying to give you a flavor for the kind of patterns that we can pull out of this. And I'll come back to sort of what we hope to do with that in a moment. But I hope that was a sort of brief demonstration of some promise. We're still early in this work. We have a preprint up, but still working on hammering out some of the finer details of this. But I think that there's some promise here for using these wavelet transformed allele frequencies to both characterize geographic patterns and relatedness and identify specific loci that are locally adapted when the processes that drive these patterns may change from place to place, such as patterns of gene flow and patterns of changing selection, and they might be acting at different scales. So I'm going to move in the last part of this talk now to try to think about whether we can learn anything useful about the genetics of local adaptation in studying land races of crops. So the sort of challenge is taking these genome scans like I was describing a minute ago and applying them in some useful way to leverage knowledge of local adaptation. Now this is something that I was sort of contrasting my previous approach, the wavelet tests. I contrasted those with this genome environment association approach, but this is another separate approach that we also have been employing. And in this case, if you're not familiar with this, what people do is you go out and sequence populations from different locations and then scan the genome for a particular loci where you have an allele frequency correlation with environment. And that's putatively a pattern indicative of local adaptation. Now, these genome environment associations, people have been doing these for a while now. And, there's been many methods published for these genotype environment associations and many empirical implementations of them. But one thing that I think is still missing from the body of work from the community, and this is missing from my work as well, so I'm also guilty of this, is that we have had very little experimental validation of the top loci that come out of these genome scans. So it's still sort of up in the air whether when these genome environment associations get implemented on real data sets, if we are pulling out the true locally adapted loci from those. That is at the individual locus level. Now, another way we might implement this genotype environment association tool might be considered what we're calling a reverse ecological genetics approach. And so the idea with this approach is to take some known variant that has some potentially ecologically important function, but where the variant is known to actually have a functional effect. So in this case, we're taking work from Murphy et al., where those authors from a linkage mapping population, some subsequent work involving crosses and complementation, they showed that this gene that's involved in the circadian oscillator has a loss of function allele that underlies an ability for sorghum to flower under long days. So this functional allele here is responsible for the plants that require short days to flower, and they won't flower under long days. And so given the latitudinal range that sorghum occupies, given it's found in places where they have long days and places near the equator where it's always short day or always about 12 hours of sunlight a day, you might think there's some strong spatially varying selection at this locus. And so we went in and looked at the distribution, going from the result that identified these natural variants, then went into the population genomic data to ask whether there's evidence that these are locally adapted. And so we see this strong latitudinal decline in allele frequencies with these two alleles, where the photoperiod sensitive allele that requires short days is more common in tropical latitudes. When you get to the temperate latitudes of Southern Africa, the allele that is able to flower in those long days becomes dominant, suggesting that this locus is involved in local adaptation. Now, I'm going to tell you a brief story here about a different trait and a different gene that I think illustrates maybe some of the potential promise for using this knowledge from the genotype environment association. So this is Astraga hermonthica, that's the pink flowered plant. This is a root parasitic plant that attacks cereal crops. And it's a major problem for smallholder farmers all across Africa. It causes billions of dollars of losses when you aggregate across crops. And one of the things that we got interested in from the perspective of local adaptation is that perhaps we might see local adaptation to the parasitism of Astraga, because parasite and host are both native to sub-Saharan Africa. But the host has a much broader distribution than the parasite. The parasite is a bit more restricted to tropical Africa, whereas the host makes it into the temperate regions. And we were wondering whether if there are any resistance alleles out there, if those might be involved in local adaptation, or tolerance alleles for that matter. So the idea is that there would be some genotype environment interaction for fitness, such that the genotypes that are adapted to the high Astraga regions are able to avoid parasitism in some way. Maybe they're resistant, but that those where Astraga is not present may have some other adaptations to the absence of Astraga. So this work kind of took off from this paper by the Jeta lab at Purdue, where they identified a natural resistance allele at this locus in sorghum that impairs the germination of the parasite. So this is a sorghum root. These are the parasite seeds that are not germinating in response to this resistant host. So they don't know the host is there, essentially. And in this case, the host is stimulating germination of the parasite seeds. And eventually those parasite seeds will germinate, or they'll continue to grow into the host roots and take out resources from the host, damaging the host's yield. Now this gene has the effect of changing the kind of hormone that's exuded from the root into the soil that the parasites use to sense the host. Now, what we were wondering though is, the study that published this identified these natural variants from a biparental cross and did a lot of the experimentation in the lab. One field site, I think, was involved. But sort of the broader question that I think we were able to ask with some of the population genomic data is, are these resistance alleles useful in a variety of contexts? That is, a variety of environmental contexts, because there's all this high dimensional environmental variation out there across Africa. And also, perhaps, is this resistance allele useful in a variety of genetic backgrounds? Because there could be some potential negative interactions with certain genetic backgrounds. So this is what we wanted to do, was ask whether there's evidence that this is involved in local adaptation of the parasite. This is work that was led by Emily Bellis, who was a postdoc in my lab a couple of years ago. And what Emily did was took observed distributions of the parasite and environmental layers and built this model that's essentially continuous surface estimating the prevalence of the parasite, or the habitat suitability of the parasite. We then wanted to use this as our environmental variable to test for a genotype environment association. So on this plot here in the middle, I'm showing you the map of Africa, where the underlying surface is the prevalence of Astraga, or the suitability for Astraga, where green means more Astraga, which is their parasite. And each circle is a sorghum landrace. And the circles are colored according to whether they have the intact susceptible allele at this gene, called LGS1, or whether they have the deletion allele, which is the putative resistant allele. So one thing to note is that the resistance alleles in purple are distributed in West Africa across a pretty broad region. Some places in East Africa where you have sorghum or Astraga pop up as a problem, in southern Sudan, a little bit in Ethiopia and Uganda. And so that does suggest there is at least some potentially broad utility for this deletion allele. It's found in a number of different places where the parasite is found. We also see in this is a neighbor joining tree of the land races. There's a few different putative resistant alleles that I've sort of collapsed on this map, but they're segregated here in coloring whether the landrace has those alleles or not. And you'll see that although there is some clustering here among genotypes that are related, they are fairly broadly distributed, suggesting that they're not just restricted to a single genetic background. So that's potentially a good news for deploying this allele across more diverse material. One cautionary note, and it's another way to sort of look at these data, are plotting the allele frequencies of this locus against the habitat suitability for Astraga. So you do see this fairly strong and statistically significant pattern of increased resistance allele frequency as we get into regions of more Astraga habitat suitability. But I think it's really notable that even where Astraga is potentially the worst out here, that resistant allele never dominates the populations. It's always at a modest frequency, including in this region in northern Nigeria where it's the most frequent. It never gets to be more than 30% of the land races that carry this allele. So that suggested to us there might be potentially a trade-off or something counteracting increase in the frequency of this resistance allele. And set that aside for just a moment, because what we also did was deployed that environmental gradient in a genome-wide association scan for allele frequency versus Astraga habitat suitability. And we have some candidate loci that are highlighted in the colors there. I'm not going to focus on any of those. One novel locus that I'll point out is a pretty high frequency SNP in a pectinesterase. This is a gene that modifies cell walls, which is of interest because the parasites and the hosts have sort of a tug of war going as the parasite tries to penetrate the host cell wall. And there's also a peak in Jajima's D at that locus. Look at the allele frequency of that thing across the landscape. It has a pretty good sort of mirror of where Astraga is most prevalent. So we are now finally starting to do some single locus validation of these genome-wide scans. It's a little late, but we are doing this now in sorghum in collaboration with breeders. Now, but I want to get back to that sort of reverse ecological genetics approach, starting with that known functional variation at LGS1. One of the things that came out in the initial paper that characterized this locus was that the authors conclude that it results in quantitative and qualitative changes in the stragolactones, which are those hormones that stimulate Astraga germination, lowering the Astraga germination without negative productivity side effects. And this last part that I think is a little premature, given some of the pieces of evidence that we have from the allele frequency patterns. Namely, if you focus in on various regions and you sort of look at the edges of those regions where Astraga disappears, the resistance allele also disappears. And you have that sort of at both edges of the regions where Astraga is prevalent. Sort of these resistance alleles don't really go into nearby areas where the parasite is absent. And you can sort of see that in this plot as well, where it's extremely rare, where the parasite is really absent. So to us, that suggests there might be a trade-off. And a little bit of a hint about that trade-off is that this specific stragolactone that the resistant sorghum exude that doesn't stimulate germination of the parasite in this, it's resistant in this study. It's also the same stragolactone that rice carries, which mainly exudes orobankol. That's the same kind that was resistant here, but rice can also be parasitized by the same parasite. So that to us suggests that there are populations of the parasite out there that have alleles that make them capable of sensing this putative resistant hormone. So we're following up on some on that with genetics of the parasite populations from different regions. Now, we also benefited in this study with collaboration with Corteva Agriscience, which is an agricultural company, who made CRISPR deletions. We were on GAL specifically, made CRISPR deletions that mimic the natural loss of function variant at this locus. And we tested those CRISPR lines against parasite seeds from two different locations, one from Mali and one from Kenya. And so here on this figure, we're showing you the Astraga germination results. So low germination means resistant, high germination means susceptible. We did this in three different environments. And so one message from this is that the deletion allele always has lower Astraga germination. So that confirms there is some resistance conferred. But it's important to note that this changes across environment and Astraga genotype. In particular, the kibos, the Kenyan genotype of the parasite, germinates at a reasonable level, depending on how many seeds are in the soil. This may be a number of Astraga individuals that have germinated, even though it's still lower than on the exudate of the susceptible genotype. So that suggests that one potential trade-off or something limiting the frequency of this resistance allele could be the potential for the parasite to evolve in response to increased frequency of this LGS1 locus. And then another thing we want to do to try to understand potential trade-offs associated with this resistance allele was to characterize transcriptomes of the CRISPR lines and to get at how the rest of the plant might be changing. So this is done in the absence of the parasite. And stragolactones, which I didn't really tell you before, are hormones, and by definition, they influence a lot of different processes. And they are synthesized in roots, transported to shoots, but also exuded into the soil. So anyways, we found lots of genes that are differentially expressed in response to this single deletion of this LGS1 locus. So there's some pleiotropy here. And the strongest functional enrichment that we see with this transcriptome study is in the shoots, where we have a downregulation of photosystem genes. And this is consistent with some work in tomato. It hasn't been addressed in sorghum yet, although we have in a moment here, I'll show you, where they show that these stragolactones positively regulate light harvesting genes. So it could be there's some impairment in photosynthesis that is a cost or a trade-off associated with this LGS1 deletion. And we see some evidence of this in some greenhouse studies of the deletion lines and looking at their photosynthetic rate. So this is across a range of carbon dioxide concentrations. The susceptible line has higher photosynthetic rates than the resistant line. And you can just average across, or this is sort of looking at a normal CO2 level. There's a lower photosynthetic rate of those resistant alleles, even though there's no visually obvious phenotype. So that suggests there may be some subtle trade-offs that may be enough over long periods of time to limit the distribution of this resistance allele. Okay, so to conclude that part, wanted to show that there's good evidence that this gene that controls stragolactones is involved in local adaptation to Astraga via resistance, but there may be potential trade-offs that could limit the utility of this allele if we were to deploy it in sorghum breeding programs all over the continent, let's say. I'm not saying that it's totally worthless, because farmers are kind of desperate for any genetic resistance to this parasite because they often lack the resources required for other methods of control. And then secondly, there might be still some promise for using these associations to find new genes that are involved in local adaptation. Okay, thanks everybody for hanging with me without presenter mode. And I want to thank my funders at NIH and NSF, who also gave fellowships to two postdocs, or Emily Bellis is now PI, and Erica Lawrence, who's a postdoc currently, and then a number of other authors who assisted with collection of material or experiments on the Astraga sorghum resistance work. Thank you all for coming and listening.